This All right, guys, this is going to be one of the shortest videos ever, I think. We're going to talk about facilitated diffusion. And so, diffusion, just because we haven't clarified it yet, is the movement of molecules down their concentration gradient. What that means is that they are moving from a high concentration to a low concentration. So kind of like what you're seeing happening here. We had a high concentration on this side. We had a low concentration on this side, and we had a net or total movement of molecules from the high concentration to the low concentration. Now, this actually should have moved one more molecule over because these should move until they reach what's called equilibrium. Which is when the concentrations on both sides are equal. So this happens to small nonpolar molecules, and those are the molecules that are able to move from one side of most membranes to the other side. And keep in mind, when we're looking at various things, we're looking at these things in aqueous solutions because they're in biological systems, and we're looking at membranes that are probably going to be made of phospholipid bilayers, and we'll talk about a few other things later. Facilitated diffusion happens when you need to move something that the membrane is not permeable to. So the word facilitate actually means to help. It's diffusion, so things are still going to move from a high concentration to a low concentration, but it is going to be with a little help because you have to have membrane proteins, either a transport or a channel protein, to help move these things across the membrane. These molecules will move down their concentration gradient, meaning they'll move from a high concentration to a low concentration. And keep in mind, there is no energy input, so this is a form of passive transport. So some examples of some of the commonly moved substances would be ions like sodium or potassium if you're trying to move any other type of ion like chloride or calcium. Large polar molecules, things like glucose, will also need to be moved this way. So let's talk in particular about the circumstances of sodium. So sodium and its concentrations across a membrane are really important for function of the nervous system. And what happens when you have these ions at different concentrations across a membrane is you can actually cause a voltage difference. So this gives membranes a charge, or what we call a potential difference, and that means the membranes are polarized. So notice here, where do we have more sodiums? We have more sodiums inside the cell than outside the cell. That means that we have a more positive charge inside the cell, and that means we have a difference in charge across this membrane, which we define as being a polarized membrane. So we won't go into this in more detail because we no longer cover a lot of details about the nervous system, but if you can move these charges across the membrane, build up this concentration gradient and this potential difference, then you can actually conduct an electrical signal down a nerve cell, and that's actually how nerve cells communicate. And then the other super big example of facilitated diffusion is going to be our aquaporins that we've talked about in class several times. A very easy way to remember this is to remember that aqua is very similar to the Spanish word for water, agua. Aqua is also the color of, blue that's supposed to be like water blue. And then if you think about it, a pore is like a hole in something. So this is literally a pore for water to allow it to cross a membrane freely. So most biological membranes are going to have plenty of aquaporins to allow the free diffusion of water across the membrane, which is going to become very important for our next lesson, which is going to talk specifically about how water moves across membranes. All right. That is it.